to the topic 7. This is my last one for unit 1. So we're gonna talk about membrane transport. And yeah, it's already 9:15. Membrane transport. So cell plasma, there's a hydrophilic side, hydrophobic region, and another hydrophilic region. And the function of these homeostasis and the membranes, well, there is the separation of inside and outside, the homeostasis of maintenance of inside and outside, selective permeability, meaning allowing some in and allowing some out. And the whole point of this is to be a selective permeability, so that they controlling what's going in and out. So this is a diagram for, what is this? Yeah, this is just simple diffusion. This is called simple diffusion right here. I already wrote it. So small hydrophobic molecules, usually gases like O2, CO2, N2, benzyme fats, can just come in through the membrane. This is just membrane, imagine membrane, really easily. Small, no charged polar molecules. So if they're polar, but they're small, and they're no charge, then some of them can get in, some of them, but some of them won't get in, such as water, glycerol, uracil, and sugars. If they're large, not charged polar molecules, so they're not charged, but they're polar, and they are bigger molecules, then they are a lot less likely to be able to do the simple diffusion, because they are they're hydrophilic in this case, instead of being hydrophobic like this, right? Because these ones are hydrophilic already, and these are hydrophilic, and hydrophilic is just unlikely. Ions are completely NG. They will not simple diffuse at all. That is why there is passive and active transport. So passive transport is not requiring energy, and they're usually facilitated diffusion, meaning there is some devices that can be used. And this passive transport is only for facilitated, or I should say, high to low concentration. So concentration down, going down the concentration is always passive transport, and they require no energy because it's just automatic, because it's just spontaneous. And it could be facilitated, so facilitated diffusion instead of simple diffusion, facilitated by channels and carrier proteins. And we're going to see what that is. For example, aquaporin for letting water in. So no energy is required. Keep that in mind. These are no energy required. These are diffusion. So simple diffusion is the first one I talk about. I'm just going directly. Facilitated diffusion, channel and carrier. Channel is the hole. It's a tube like this. It's just going like that. And ions can go in, right? Carriers are like this open stuff that can go in, and then we'll shut it and it'll open over here. Imagine it opens over here and it just goes in. And these are some specific ones because they can let in big insoluble solutes in actually no, these are all down their concentration. Yeah, so these are still not against the concentration favorable stuff. Water, protein, sugar, salts can get in. Water, protein, sugar, salts. So one chemical binding of substrates causes the shape change in the transport protein, and these are the transport proteins. No outside energy is required, whereas active energies, they do require energy investment, and they're usually against the high concentration. They will go concentration up, so from low to high concentration of something, that's very unfavorable, so that requires energy, and that energy is ATP in this case. And they will require carrier proteins, carrier energy against this concentration. So coming back to the simple diffusion, we just talked about that. And remember that the concentration gradient means that any higher level concentration will want to go to the lower concentration. That just happens. So carriers are the ones that are open and closed. Channels are tubes. Remember that. So, unlike lipids or O2 or CO2, which can simply diffuse, water and these protein molecules, these are polar, and that's why it's not easy to get in. That's why they need to go through carrier proteins and channel proteins. And these are based on size and ions, whereas carriers are based on binding, so if the substrate is able to bind or not. So osmosis is when there is a semi-permeable membrane in between and draw it, but there's a U-tube like this, and there is a semi-permeable membrane in the middle where only the solvent will go. So solvent will go towards the more higher concentration, only the solvent. And it's semi-permeable because it will select what can and what cannot pass through. And these membranes have special channels. So these are channels. On these membranes, there's a special channel called aquaporins, which are channels like this. Oh, sorry, channels are these ones. Channels that let water pass through and nothing else. And yeah, osmotic pressure, and etc. This one we need to memorize. Hypotonic is when the inside is too dilute compared to the out. Actually, it was the opposite. Yeah, so hypotonic is when the outside solute. So solute is higher inside the cell. So if outside is more dilute, then water will want to gush in and then will explode, which is good for plants because they have turgor pressure, meaning turgid or rigid, tough state instead of lysed state like animal cell will explode. So plant cells are able to expand because of the cell walls. They're flexible and they're able to expand, and they actually like it when they're in the hypotonic. So we can put them in water and they like it there. If it's the same outside and inside concentration, it's isotonic and nothing's happening, it's just normal. And these guys are going to be called in a state of flaccid state. I guess like a neutral state. And solute, if the solute concentration is high in the outside, so if the inside is more dilute, then the water will gush out and they will be shriveled. And that's a hypertonic. Hypertonic is when they gush out and they are shriveled. And plants, that's disastrous because that's a plasmodized state. A very unfavorable state because they're just basically dried out. Osmosis and diffusion. So simple diffusion is when the solutes move. So diffusion is solute moving from higher concentration to lower concentration. So you may imagine beaker, there's a wall, and you take away that wall, so then it's just a whole beaker, and the solute will move towards the less concentrated. So the higher concentration will want to go to the lower concentration. Osmosis is only the solvent moving. So now let's talk about the active transport. Why can sodium diffuse into the cell? Because it cannot diffuse from lower to higher. So sodium usually in the outside is a higher concentration always. So sodium is always a higher concentration of sodium outside. And if you want to get sodium out, then that's very hard to do because it's against the concentration energy, right? So then you need to invest some energy in there, which is the ATP. So you require ATP. You need input of ATP.
Are you recording?
Yeah.
I love it because if you're hearing this, okay, just reminder that
published. So the ADP and ATP. There is the turning ADP into ATP, which is done in mitochondria, but for now, so adding phosphate is adding energy, and removing phosphate is hydrolysis, is releasing energy. So by adding a phosphate, you're adding energy. So ATP can be used for
where's the AMP
reaction as well. So there is 3 directions. One is only one direction. There's both direction. Another protein is towards the same direction, but 2 can go in. So now we have inactive transport that require energy and is against the concentration. There is primary and secondary and bulk transport. So primary is when they use energy directly, so ATP is used directly. So adenosine triphosphate is used directly. So hydrolysis to to release the stored energy, and every time the phosphate is added, there is more energy, and every time the phosphate is broken, there is a release of energy. Across membrane is glucose, amino acid, nucleotides, and salt. And these moving of ions are what makes the electrical gradient. So the electrical gradient is basically the concentration that gushes into a lower concentration used by the nerve cells to conduct muscle contracts and used for secondary transport. So electrical gradient just means a very high concentrations of ions. I'm assuming.
I love you, Pickles.
A worm? Oh no. So an example, really a major one for the primary is to use ATP directly for the sodium-potassium pump in animal cells. So what they do is to pump out sodium using ATP to gush in glucose, which is against the concentration. So glucose is really big and really hard to get in, but we need to put them in. So what we do is to pump out lots of Na outside, so that's 3 Na outward, and 2 potassium will come in. So then the outside will be more positively charged, and that is going against the concentration. So the concentration is higher outside. So then what I can do is to cause a secondary transport, which is over here, and the energy from this electrochemical gradient generated by the primary that we just talked about here is then used for the unfavorable reaction, which is the bringing in of glucose. So glucose is still against the concentration, but however, the HA, now that it went all outside, is going to be going down the concentration. So it is energetically favorable. And we say that it's secondary because it is using ATP indirectly. So the secondary itself, so bringing in glucose itself, is indirect use of ATP because it was it was first initialized by the primary to make the electromechanical gradient situation for the secondary to happen. And they use co-transporters called the carrier, antiport, symport. What is that? So yeah, antiport and symport are the forms of secondary transport that moves across cell membrane using energy.
Symport is both in the same direction.
Okay.
Antiport is in the opposite.
Okay, so there's 2 types of the input or output. So the antiport is sodium and potassium is antiport, and sodium glucose coming is symport. And bringing in essential sugar and amino acids using these kind of techniques. So now talking about the bulk transport, which is my favorite transport, is the vesicle transport, because the vesicles are containing lots of stuff, very big molecules even, and what they do is they require ATP, of course, because they're primary type, and they require ATP for all of these. First is exocytosis. Exocytosis is sending off molecules using vesicles, secretion of protein, releasing neurotransmitter, turnover of plasma membrane. So any vesicles that are getting out of the membrane, that is exocytosis. So stuff are getting out, releasing neurons and stuff. This is active transport, of course. Active transport. So don't forget that active is all of this. And there is also endocytosis, of which there are specific ones. Phagocytosis for eating big stuff, molecules. Penocytosis for drinking, so liquid coming in. And receptor mediated, meaning a specific protein that will ask for the specific endocytosis to happen. So highly selective, energy-dependent mechanism where cell initialize specific macromolecules such as hormones, protein, and nutrients, binding them to surface receptor. The process involves the ligand receptor binding, clustering. Anyways, it's a very specific case.
Okay.
And that is everything.
Okay.